think this was also real. And Sanity have, like most recently that.
All right, how is business up in Bellingham?
Stone, but like if you don't, you know, really do, it's your outlet, yeah, so sometimes,
Uh,
you know, a little bit.
Fine, I was,
Yeah.
down in Carmel, and then recently got back from Wichita, Kansas, where her family lives. It was her older sister's 80th birthday. So we did, all of her family was there, all of her siblings, and an unexpected snowstorm, about 10 inches of snow that a slog through. Yeah, fortunately, it's flat there, there's no mountains, I think, between New York and LA. Or no, but there are some, but there aren't any in Kansas. Yeah, there aren't any in Kansas. Yeah, you've been in touch with your mom and dad recently?
So then do you work with genetic counselors then? Parents
Yeah,
had to test.
Thanksgiving.
We're very lucky to have a genetic counselor.
I get in touch right now.
And a multiple
Do you like
rheumatology
I'm gonna see how you do it.
and we have a genetic counselor as well.
Okay.
So I actually
Must be pretty happy about that.
I was involved in that clinic early on in my career. And it's probably how I started to fall into like genetics and because we take lots of so it's getting more and more kind of testing, trials on
Okay.
prevention like stains.
Okay.
So the caveat, I mean, you've heard it's not much different than the talk I gave,
Okay.
but and yeah, it's really interesting.
Did she think like how you were not?
You
That's more from.
it's a lot of times I mean, probably would say this, right?
Yeah, like
Half the time it's like truly like a hallucinogenic or pathogenic, variant, but like equally enough, it's the US that you have to like do for anyway. And so, it's really be very frustrating and super time consuming.
And we have.
yeah, and then that's on top of already. I feel like there's on behalf.
Yeah,
They're nervous.
And I think that's actually maybe one reason why there's not as many immunologists as there are allergists, because
And then we.
I mean certainly like community immunologists will in like 36 stuff.
Yeah.
But
So
to really be an expert in
Verification, that's not.
immune deficiency, inborn allergy immunity, you have to see a lot of patients, and it's hard to justify like from a billing standpoint the amount of time that actually takes.
Sure. Yeah. Okay, I supported
Yeah, so.
negotiated disease advisor.
Yes.
Of course, yeah.
And a lot of those providers, so I think probably it's not your choice. A lot of those, and just where I at a particular or center of excellence, so they're paired with like, oh, I have a surgeon.
Yeah.
It's the same calls, and then that has become a sort of end up hanging work out like diagnostic and
Yeah.
into like
And it's an interest.
it's
Yeah.
efficient model that like
Yeah.
yeah.
Yeah. I mean, yeah, you can see the volume of immunology patients as so I mean I see sociopathology immunology.
Okay.
The like I still doing a true in the field of immunology. That's what I do.
Yeah.
Control the corners of the world, like that's all they do. But then they also have research and
Last year.
yeah. So
It's a lot.
it's
Yeah. Okay.
I couldn't.
And then we.
There's always a push in academics to like hyper focus in one area, right? To be an expert in that area, but the struggle, because I get so much different. I get a lot of different benefit and where I was saying satisfaction out of immunology patients than I do allergy patients, right? Like so, food allergy, it's very education based. Like we're doing a lot of teaching the families, teaching the patients. You make huge changes in their lives by like helping mitigate anxiety, like shooting diagnoses that were maybe made incorrectly, or like, helping like a side effect outcome an allergy, things like that. And so those, it comes all the time, but it's not, it's a clinical aspect of that, it's not complicated, right? The immunology side of things is where you get like all of them, like trained, get it puzzled. Because, like, find make a diagnosis, it's a huge win, and it's really life changing.
There's so many variants.
Yeah, so many areas of thinking.
Yeah.
Yeah.
So good.
I mean, it's hard. Well, it's where I struggle is to try to do both. Like, staying abreast in all that's going on in the space and try to stay in the space. What's wrong?
And like it's junk and I like to.
Yeah.
And a dog.
So we think about you again, we're not being genetics in these fellowship, like how do you see that out?
Say about it so hard.
Yeah,
Like we have a different so she worked with now on the research side with her. So I'm one of the like involved, and we have a research based culture call where we do whole gene, we do issue based assays and things like that. And so she's like on research and things, but she was initially the disease counselor in that business study. So she took some stuff, self-educating, reading stuff, just listening to like how they do it, implementing it, see what works, see what doesn't work. Things like that. Some stuff like recently we had a geneticist from Invitae.
Okay.
She's higher up in Invitae, she's not just a geneticist, but her name is Britt Johnson. And she gave a talk on, it wasn't just on today, it was actually on AI. Like it covered a lot of things, but like part of the talk was on artificial intelligence and sort of the advancement that has had in silico testing and like the benefits there. And because like their whole big thing in NVDA is to try to artificial intelligence, machine learning, and deep learning. Like, use it's about 50% of results are the US's, right? So they're trying to think that so that because there's no way you could functionally validate all of those US's. And so they're using a lot of machine learning now, and it's actually been really successful. So she talked a lot about that. But
wow.
yeah.
I can imagine using that kind of, I feel like we use in pharma or drug discovery, and they do it in medicinal chemistry to find like initial hits and they it's a target and see which is such that, and they are trying a lot of machine learning to better job.
I mean,
narrowing down what's the kind of compound
yeah.
But it's the results haven't been great. It's totally random. Like then there's a bunch of things have to follow. Genetic testing at least like once if you figure it out.
Yeah.
You have
Well, if people are like, you say AI, you say machine learning, it goes determining, right? It's like, we literally use it every single day.
Oh yeah.
Anything from like, is it going to rain today? Like there's machine learning that goes, it's not just like it's no longer it's not it's like it's right. Yeah, it's machine learning.
Yeah.
So
they ought to have.
I know Amazon is pushing it for a while. They started ABC and by the time we like got order, they do started moving boxes. Looking cleaner.
Oh.
They have like some browsing things. Started moving, machines are like moving in there. Anything you can ship it to before you like,
I'm not surprised. And you start to taste in something. What we're talking about.
More delicious. Do you know one of my parents? Very sorry. Really so.
Yeah, it's fine. Sorry. Turned out because garbage truck. The streets are very small. It's really old days. So they can't fit modern garbage down that one. You know what?
What was it again?
It's because the streets are really, it's really old.
Yeah.
Can't fit.
Oh, really?
Like, so manually people help to lift it into the garbage truck and so. But with the. So quickly, yeah, I can't remember guys. Yeah. Or yeah, 12. Yeah. We must be similar. Yeah, I don't know. I didn't even question like, yeah, just.
Yeah.
We should shut up. Stop here. Oh my god. Dying and coming out. So my back, she goes, like my knees. The ball around, like for a strong wall and stuff, to like share contacts and stuff, or to like turn it, so we do it, just. And which is kind of weird anyway.
Yeah.
Yeah, to bump up to any.
Yeah.
Yeah. It's morning. That's my famous. What's on
It's virtually, I think. Well, we met in person. That's right. I met him. We were initially going to do. We were. This is like.
Yeah.
Is he the Ohio person? Or
Yeah, it's John Jess. You guys see my 5 years out. We're done.
So I think I met some person as well. Like not and see. Yeah, came up and we already he was going to be in town. So we just he was going to
Well, it's that good.
Yeah, I've been to West Coast. Well, I don't know, I guess you consider this West Coast, but was in Utah not that long ago. I just was in Montana in October or so. I've been out here multiple times. Utah was yeah.
Yeah, he was there. Yeah.
So I gave a couple talks there. One of them was a similar talk to this, and then in Montana was a
Yeah, hopefully us and the sterners.
yeah.
So.
It's still long.
I haven't seen my.
Points out here.
Oh, no, it's very. Well, that was too. Where did you guys get mad?
Off was doing Chicago, so we threw down.
Okay. So will you fly out of New Jersey?
See how it's called out.
No.
Oh, there's only one. So yeah, it's actually wrong. No, he really couldn't. Basically, I think you're through on it. It's just like, yeah, so if you want to go anywhere. So,
Yeah.
like a complete jerk when he smells it.
Sure. I didn't realize and then email, but I said, children's medical language.
Yeah.
Right there, person. I was like, maybe I got into the right color.
Yeah, well, apparently that's not an easy task for me.
Yeah. We had to catch this guy coming up the stairs. I was like, are you my stuff?
Yeah. So easy, yeah.
That was from Dr. Altman. Good morning. Hi, Dr. Altman. I haven't met you in person.
Right.
I'm Lauren. He's the faculty at Children's.
Yeah, your name came up last night, that, guy. Thank you for coming. Anybody else from children flying over?
We can get everybody, so they're gonna probably maybe call in. But hopefully some people make it to a whole room now.
She's one of them.
Yeah, she doesn't have some kind of experience. Yeah, I'm so not too far. Yeah. I do now. What? About 11 weeks, and now they had doing so that's. Next week. Today, it's really easy and then just a couple hours. But it's free with our. So I can.
What's that? Yeah.
And it's kind of the main office.
Yeah, we were just talking last night, I mean, in person is. What about, I think it's to just talk to one another and shake hands and look people in the eye. There are nurses that accomplishment. Learn things, I can hear. You might hear something. That is exciting to study. The fellows from this board group, but you can use the lobby or the linguistic room. Can you say?
Involved by all thoughts. We'll figure it out. I was gonna say, I won't. I know, can I sit by you?
Yeah, when I was at Metanews.
How'd you think it's doing?
Right, is it on screen?
Oh. That's because nervousness.
More fellow.
Big, right?
Oh, you're some. Let's see.
That's great. Yeah, I gave an ice flutter.
Here it's the same. The seating show.
Wow.
I'm not taking pasta on my plate.
That's my mom.
Right now.
That's all you're doing. I've never seen sort of.
Looking for that.
And there's no baby ice flowers. So, for fun.
You've been calling this study. 3 is probably the last.
Another tier, mine would come back. But my head's in Edmonds, which is super nice. And then mom was all there. Like get a Christmas tree. It just didn't work for us. But we're actually gonna have one delivered there. But yeah. And it's not half, we're trying to drink the water.
Where are your colleagues?
Yeah.
Sleeping? Yeah, you're not responsible for them.
Like, oh, I'm coming out. No, I'm not. I have to go for it. That's for me. To see the have to do some garage. So I did, and I got like there, scattered. Yeah, I didn't realize it was up the slopes. I was trying to get up the elevator. I don't know how it works. Very to get touched. Yeah, it's getting time. Don't be going. It's not much for doctor. They just like push the door and like for two elevator. Oh, you're almost you go much. And I can take it down. Here's that always. It's quite the street here. Awesome.
I can't.
I have to say,
Everybody. Started. Dennis. All right, well, it's nice to be back in person here. I hope we can run more of these sessions in person. I think people still enjoy looking at one another in the eye. We've been talking about that at dinner last night, and all kinds of topics come up that don't happen on a Zoom conversation. Very happy to introduce Benjamin Prince is here from Ohio State. I wonder why you call it Nationwide Children's Insurance Company. Nationwide Insurance.
Yes, do you know what our ER is from the street?
Go ahead.
Do you have a convenient ER? All brands is based in Sutton, so there you go.
Coming it down here, to give us a presentation, I'll just read it here, of all interpretation of genetic things. It's an entirely new thing to me. I know an awful lot this one, the diagnosis and care of a immunodeficient patient. So, a pleasure to have you here. Thank you very much.
All right, so I'll try to talk to you loud in this. Pull this back a little bit. So it truly is a pleasure to be here today. Talk to all you guys. Be a part of this very well-known didactic series. Meet Dr. Altman in person and meet all you guys last time. We had a great time. So as Dr. Altman mentioned, I'm going to talk about the role in rule for and interpretation of genetic testing in immunodeficiency patients. Let's see. Here we go. Here are my disclosures, which should not be relevant. Yeah, we are. All right, and here are my objectives. But essentially, so my goal for today's talk is we'll do a quick background on just genetic testing in general and variants that you might see. But I'm going to spend the majority of the talk, the good second half of the talk, talking about what do you do with those results when you get them. And so I was told to incorporate the word of the day, I think for CNE purposes, which is genome, very appropriately. And I like to start with a case. So this is our first case of the day. So you're seeing a 10-year-old male with central adrenal insufficiency who's presenting now for concerns for environmental allergies, secondary to having a runny nose and cough for years. The family felt that his cough was secondary to nasal drainage. He had no history of wheezing or prior diagnosis of asthma, but when you get a little bit more history, and this is actually a real patient that I saw in my clinic, he apparently had a chest x-ray diagnosed pneumonia twice. He's had frequent viral URIs. He was also previously admitted for fevers, vomiting, abdominal pain a few years back, and found to have reportedly low antibody levels at that time. They were explained that it was maybe secondary to his concurrent illness, but he was ultimately lost to follow-up and was never rechecked. No other family history of recurrent infections or autoimmunity. So I don't know about you guys, but like true, confirmed pneumonia in a younger patient, in a 10-year-old male, and this sort of history certainly warrants, I think, a little bit more of an investigation beyond just environmental allergies. So we did, and this was his immunologic evaluation. So he had a very mild T-cell lymphocytosis, normal B cells, but if you looked at his sort of the B cell phenotyping, he had low classic switch memory B cells, and then most markedly, he had a pan-hypogammaglobulinemia and had insufficient or suboptimal responses to both protein and polysaccharide vaccination. So what do you guys think? What would his diagnosis be in this situation? You guys can throw it out. We're gonna have some audience participation today just because we're in person. What would you call it? What is this?
CVID.
I agree, CVID. So it's a very appropriate diagnosis. I mean, he meets the criteria, which I like to call sort of an umbrella diagnosis based on laboratory criteria. But I want to ask you a question. Would you send genetic testing in this patient? What do you guys think?
One of the fellows answered.
Sorry, how old is he again?
He's 10.
Oh, probably, yes.
Probably yes. All right. Well, let's postpone that conversation and do a little bit of that. So I think you guys are all very familiar probably with this slide. This comes from the most recent IUIS publication from last year. We're now up to almost 500 defined inborn errors of immunity. And this paper, they classify it based on immunologic phenotype into 10 different categories. They're broken down into tables. It's a really nice reference if you're wanting some quick information. The reason why I incorporated this slide to start is what's really striking, I think, about this publication is really the exponential increase in new diagnoses of inborn errors of immunity over the last 20 years. And I think what I want to point out is these are not these are not new novel diseases, right? These are diseases that probably many of them existed before. But what's different is really the advent of genetic testing. So over the last decade, we've learned a whole lot about genetics in general, as well as the interpretation of the different variants that you might see. And then more importantly, the access to that genetic testing has been much more tangible, and even community allergists and immunologists are sending it. It's much more affordable to the patient, and so it's really been a huge litmus or impetus for like the expansion of novel diagnoses in this category or in this space. And so there's a lot of things that genetic testing is going to be useful for when you're seeing patients with presumed inborn errors of immunity. In addition to providing a specific diagnosis. So actually narrowing down why are they presenting this way. And I will tell you, I have been fooled. So oftentimes you might go into the genetic testing thinking that this is probably a result of this, maybe a variant in this gene or that gene, but many times our patients don't present like they are in our favorite immunology textbook, right? So you can have hypomorphic variants and variability in expressivity that can sort of fool you. In addition to that, it can provide other benefits to the patient. It allows for precision therapy. We can have enough biologics and immunomodulatory medications that allows for anticipatory guidance and screening. So looking for other comorbidities that we know are more likely to present when you have a defined genetic diagnosis. It's for genetic counseling. So both for that family, but in sort of for future generations. And now with the advent of being able to do things like in vitro fertilization and pre-implantation testing, it can be really powerful for that family. But it's also useful for us, right? So we have learned a whole lot about our bodies, about the immune system, about certain pathways because of this genetic testing. And I will tell you, always does the human immune system function just like the mouse. So while murine, modeling and research is very important, understanding sort of what happens when you have a pathogenic variant can tell us a lot about what is supposed to happen in a normal immune system. And then last but not least, and this is, I think, also very powerful, is we need to keep in mind that that inborn areas of immunity are in fact very rare. So when you identify a genetic etiology, it allows you to pull those patients together in a cohort and then study them more as far as how they present various phenotypes as well as their treatments, right? So you can study certain interventions that you might do and be able to come across the best management for that patient. So I'm often asked, when do I think about sending genetic testing? And so these are some things that I've listed here. So certainly the easy things are when you have somebody in front of you that certainly has a defined combined or severe immune deficiency, maybe early onset disease. Somebody mentioned that, this is a 10-year-old presenting with clear humoral immune deficiency, for growth and development. Other syndromic features certainly can be, manifesting in patients that have inborn areas of immunity. And then there's lots of other comorbidities that are actually more the result of immune dysregulation. And I think that's what's really, I want to point out here, is if you look at all the things that I've listed here, 50% of them are not really recurrent infections, but in fact, manifestation of underlying immune dysregulation. I think when I think back to when I was in fellowship, a lot of times, we think, maybe this patient doesn't have recurrent infections, so they couldn't have an inborn area of immunity. And we know now that it's not in fact the case. So before we go further, I want to talk a little bit about genetic variation in general. And so I think you guys have probably seen a slide like this before, but I think this is a good, simple representation of the different kinds of genetic variation that you might see. So in the first line is our wild type sequence. So 1, 2 big zoo, the individual letters representing individual nucleotides that make up then the amino acid. So each word is an amino acid, then resulting in the ultimate protein there, 1, 2 big zoo. So a missense mutation is when you have a nucleotide substitution that changes that amino acid, but you don't have a change in the reading frame. The other amino acids are exactly the same. One amino acid is changed, and that's probably going to be one of the most common variations that you're going to see. An insertion is insertion of DNA material, again, keeping that reading frame, but you have a new amino acid or several amino acids that are inserted into the protein. Deletion is the opposite. Duplication is when you have a duplication of that, again, DNA that's lies somewhere else in the genome. And then a nonsense mutation is when you have either a missense mutation or a variation that occurs in a nucleotide sequence, results in an early codon that's made, and that ultimately terminates the translation of that protein. And then last but not least, you can have frame shift mutations as well, where you actually lose the reading frame. So you either have an insertion or a deletion that changes the reading frame, and then you ultimately end up with a completely different protein. And those last two variants are going to be the ones that are going to be less common than this sense, but the reason for that is because they're more likely to cause disease, right? More likely to cause problems within the function of that protein. But as I kind of alluded to, variants are pretty common. So just as in a typical, human genome, we expect there to be about 500 rare missense variants. And oftentimes it's very difficult. This is where it comes down to sort of the first part of analyzing or sort of make heads or tails of the results of the genetic testing is trying to understand, is that genetic variant, is it the result of natural selection and sort of sort of contagious that's had in a protein that allows that patient to better survive versus a pathogenic variant ultimately causes disease. And so this can be the really kind of the crux of genetic analysis. So in order to kind of help with this, the American College of Medical Genetics and Genomics published back in 2015 sort of a standardized nomenclature and categorization of these variants. I'm not going to go into detail in this slide at all, but I wanted to give you an idea of what they look at, because you will see this maybe mentioned in different genetic tests, or if you're talking with a genetics counselor or a geneticist in general. On the y-axis, there are different domains. It might be population data, functional data, the way that the variant segregates in the family. And then on the x-axis is sort of what in that domain, the characteristics of that variant would make it more likely benign or more likely pathogenic. And what you do is you sort of add these characteristics all up, and in the end, you ultimately get a readout of, if you meet criteria for it to be pathogenic versus likely pathogenic, benign, likely benign, or that dreaded variant of uncertain significance when you don't have enough characteristics to push you one way or the other. And so before we go further, let's talk a little bit about the different kinds of genetic testing you might consider when you're seeing that patient in front of you. Does anybody know who this is? Any guesses?
Is it either Watson or Crick?
Oh, that's a great guess. This is actually Frederick Sanger.
Oh, the Sanger technique.
Yeah, so this is Frederick Sanger. He was an English biochemist, and he was awarded the Nobel Prize not once, but actually twice. First in 1958 for determining the structure of insulin, and then next in 1980 for discovering sort of the sequence of nucleotides in DNA. And what he did back in 1977, and it's actually still done today, believe it or not, is he took the template DNA, he denatured it, and he added in DNA polymerase, primer, nucleotides, and then several different high-deoxynucleotides. These are nucleotides that were fluorescently labeled and were lacking a hydroxyl group. So ultimately what would happen was then that was incorporated in the sequence, it cuts termination of that transcription. And so if you do that multiple times, you end up with several different lengths of sequence of that initial template DNA. If you run that on a gel, you'll then know which is the smallest sequence next to the largest, and then you can then look at that fluorescence to determine, is it an A, a T, a G, or a C? And so he was able to then determine sequence of DNA in that way. As you can imagine, if this is how we did all of our genetic testing, we would not have had the discovery that we've had that I showed you on that first slide. But it still is very, very reliable and is not very error-prone. So that's why it's done today when you're wanting to confirm certain things, or especially from a research basis, identify truly what the sequence of the DNA is. Today, we use lots of other different kinds of genetic tests, and I wanted to just take a minute just to talk about the pros and cons, because I think these are important when you're seeing, again, that patient in front of you to decide which genetic test might be the best. So I think you guys all know about a microarray, but a microarray, what it's doing is it's using probes to anneal to, again, your template DNA, and it's looking for large losses or gains compared to a reference, in an effort to identify copy number variation, right? And so depending on how many probes you have, depending on the size of those probes, it's going to affect, how the detection rate of how small of the detection of losses or gains you can find. In general, you're going to still miss small copy number variation, and you're not going to know anything about the sequence. This can be very helpful in patients that have consanguinity to determine where areas in their genome they have homozygosity or the same genetic material. But again, outside of that, and I will say copy number variation has been now shown to be very important from the standpoint of different immunologic diseases, but it's not going to tell you anything about sequence. So the next thing that you could consider sending is a small gene panel. This is if you have maybe a patient that has a very specific phenotype, right? And so there's maybe just a variety of genes that could present in that way. An example of this would maybe be SCID, right? So we know that there's identified, 20-some genes that are causal, have been shown to be causal for SCID. And so you could consider sending, if you have a patient that meets that criteria, a very small panel test. Another way that this might be used is, for example, if you have a known pathogenic variant in a family and you're wanting to see if you have other members of that family that carry that variant. It's not very helpful when you have a more broad phenotype, which is what many of our patients present with when they're presenting with concerns with inborn error of immunity. And in those situations, it's better. We oftentimes send are these more broad genetic next-gen sequencing panels. And so in this situation, you have usually a number of different genes that are known to be associated with diseases. The current panels are anywhere between 400 to 600 different genes. The pros of this test are that you usually, if the gene is listed on this panel, you can rest assured, although you can go in and look to see specifically what the coverage rate is, usually the coverage is pretty good for those genes within typically the exons, and sometimes a little bit into the intron space as well. And so you can, it's very reliable in that way. The downsides are that you're not going to identify novel disease. You're also, the more genes you're looking at, the more likely you are to find these variants of uncertain significance. So it's going to take a little longer when you get those results back. But the turnaround time is usually very good, and they can be very useful from a clinical standpoint. And patients that have more of a broad phenotype, or patients that have consanguinity, you might then consider whole exome sequencing, where you're looking at all of the coding parts of the DNA. And this can be very helpful, again, especially if you don't have a defined, you might have overlapping phenotypes that impact multiple different organ systems, for example. You can also identify novel diseases in genes that weren't otherwise known to be associated with disease. Takes a lot longer, though. And so that's not because the sequencing takes longer, that's because the interpretation takes a lot longer. You also are going to present or get back lots more number variants of uncertain significance. And because you're looking not just in your area of expertise, it oftentimes does take the help of other subspecialists to interpret findings. And sometimes when you're doing it on patients, there'll be an intake form. What do they want to know? These other maybe risk factors, cancer predispositions, things like that, when they get the results, or do they want to be blinded to that information? Last but not least, whole genome sequencing, and this is becoming more and more utilized. And one of the reasons, at least in our space, is because, I talked a little bit about coverage rate is good usually for broad next-gen sequencing panels. For whole exome sequencing, the standard backbone that is used by many companies has poor coverage on many immunologic genes that are important for immune function. With whole exome or whole genome sequencing, we're capturing the entire genome, right? So we're not missing any of that. So it's got great coverage. You're also going to get intronic variants. The problem is again in that analysis. And we don't know what, we know a lot of things as far as what's important for splicing and things like that, but there's a lot of DNA material in those introns that we're not really sure what it's doing. And you're really reliant on the interpretation testing from wherever you send it.
Maybe it's a stupid question for someone who knows nothing about this and never ordered this test, but how do we know what the normal human genome is?
So there are different, so the human genome was sequenced, I think, back, I was in college, I want to say.
Sure.
And since then, we're now on the references, I think it's H38, I want to say, so version 38, but there is a standard reference, and you have to, when you're getting your genetic testing back, you have to look to make sure that you're looking at the right reference when you're interpreting it. So, for example, if it gives you, a specific location based on, where that falls within that gene, you have to make sure that you're using the right reference DNA, the right reference, version of the human genome in order to interpret where that is with regard to that gene. Does that make sense? Kind of.
Yes, more or less. But when they did the initial genome, it was just on a few people, right?
I presumably I will raise my white flag and say I don't know quite as much of like what happened now. But I do know that again, over time we have updated versions of what the reference range is for the human DNA. A good question, actually. So this all leads me to say, well, how good is this testing in patients? And so this was actually a publication put out by Jordan Orange and Ivan Chin, and they looked back from 2014 to 2020 at any publication that utilized either broad next-gen sequencing, whole exome sequencing, or whole genome sequencing in patients that had phenotypes that would be consistent with immune deficiency or inborn error of immunity. And what they found that, putting all of those testing strategies together, the diagnostic rate was about 31%. So when you looked at patients on the left that had a higher sanguinity, you should increase your diagnostic yield, unsurprisingly, especially in the exon space. So you can see that the diagnostic yield was about 70% in those patients. But what I think is what's really interesting is when you look at all the different modalities, they're not all that different. And so I think what's important when you're considering what test you want to order, it's important to consider cost for the patients. It's important to consider the phenotype that you're seeing, as well as other family history, whether that's consanguinity or other manifestation of disease. But you're not going to really go wrong either way. And again, I think a lot comes back to the interpretation of those results. In their kind of review, they did see there was one study that looked at the yield when you add in next gen panel, like a broad panel, and there was a 12% improvement in their diagnostic rate. But again, that was one study. So we'll say how much doing Son after a broad panel is going to get you more yield. And again, it goes back to what is the phenotype of the patient.
And that first statement means that only after you do this sophisticated testing, you only help you 31% of the time.
Correct.
And you categorize that as not so bad. I would say not so good.
Yeah. So I think it comes down to the benefits of and I will say that again, this was going back all the way back to 2014. We've gotten a lot better both at doing testing, but also interpreting that testing going forward. And when you do find an answer, it is oftentimes very impactful for that patient on many different levels, actually. And I do think also that this is going to get better. And again, part of the crux of this is a lot of times we get results back that we're not sure what they mean. And so it's hard to interpret what that means. All right, so let's now go back to the patients that we were talking about. CVID patient, 10-year-old. Do we want to send genetic testing on this patient? So as I think about it, reasons to do testing would be that he does have a definitive humoral immune deficiency. He had early onset of the disease. He had this unexplained central adrenal insufficiency, which certainly could represent some form of autoimmunity or immune dysregulation. Reasons to be against testing. This is at best. CVID is you only find a diagnostic rate about 30% of the time, although we might say, well, you want them all together. That's probably what it is as well. I will go back to that study and I will tell you that when they looked at presentation, so like, again, CVID has a diagnostic rate when you do genetic testing of about 30% at best. There are certain inborn errors of immunity that have a much higher diagnostic rate. So things like HLH, cytopenias, what was the other one? SCID. Well, SCID, certainly. Well, it's not important, but the ones that have a very defined phenotype were the diagnostic rate was about 60%. So I think that there's two things that I would point out for that. So one is the more you have like a clear, more directed what is wrong potentially with their immune system, that you can look more diligently in those genes that are thought to be important. But then also it's very important to provide detailed clinical information when you're ordering the test, because whether you realize it or not, there's a lot of analysis that goes on before you even get those results. And so that analysis is oftentimes defined by the clinical information that you provide. And so the more clinical information that you have that they're looking at, the more likely that they're going to either include variants that would be plausible and not exclude them based on something that you may have left out in your clinic, in your history. Oops, this came. Minimize that. All right. So, and then the last thing about maybe not sending him is he didn't really have a family history that was significant. So long story short, I did send genetic testing on him, and you can't see it, but he had a pathogenic, it was likely pathogenic at the time, but it's now been reclassified to pathogenic variant in NF-κB2. And so what the variant that he had was actually the C-terminal of the protein, resulted in an early stop codon and truncated the protein and lost the last 42 amino acids of the protein. And so why that's significant and what he ended up having is an NF-κB2 haploinsufficiency. And again, I wish I could move that over, but so what was going on in him, up there on the left, we have our canonical and non-canonical NF-κB pathways. And so Oh, here we go. So this is NF-κB2. It normally resides in the cytoplasm in its inactive form, which is P100. And what happens when you have signaling is you have basically activation of this kinase that then phosphorylates this protein, NF-κB2, and it phosphorylates it on the C-terminal part of the protein. When phosphorylation occurs, it activates it into P52, is what then the new protein is called. And then that heterodimer of RAL-B, but could be another, there's 5 different family members in NF-κB, then translocates to the nucleus and then results in transcription and translation. So in the end part of that signaling. And so in him, because he lacked that C-terminal part of the protein, he could not activate this protein, and it just hung out in the cytoplasm. So it just sort of hung there, and it wasn't able to go to the nucleus. And so you might say, well, that's just affecting that pathway. But what we've learned is that we used to think of these two pathways as sort of, distinct, and they didn't really interact with each other. But the more we've learned about the immune system, there's a lot of interaction that goes back. It's not as simple as maybe a boss throws it out to be. And so in addition to activating signaling, that this complex, P100, the inactive form of NF-κB2, also inhibits the canonical pathway because it ultimately blocks this activation downstream as well, and kind of keeps things again in the cytoplasm. So as a result, patients that have this haploinsufficiency, they present with very classic humoral immune deficiency like our patient did. They also present with the various pituitary insufficiencies like ACTH deficiency, which our patient had, growth hormone deficiency, hypothyroidism. They can present with a variety of different autoimmunity immune diseases as well. And it's well defined now. Alopecia is one of the presenting factors that we often see in these patients. So that's ultimately what he had for this point for this part.
Yeah, because this pathway is important in other organ systems beyond the immune system.
So even if it wasn't this, so the canonical signaling pathway is very important for signaling of like IL-1, TNF alpha, a lot of the B cell receptor, the T cell receptor, they all rely on that canonical pathway, where there are other variants that have been identified in this pathway. So another common cause of CVID is NF-κB1 variants that can occur. You've heard of probably TACI, right? So TACI is in fact this molecule, is this receptor, and it's activated with BAFF, CD40, and other B cell differentiation factors and signaling molecules that help for B cell preservation. So it plays a huge role in immunology in general. Outside of this, just a small play. But as far as its implication in other organ systems, it certainly is. But the predominance of what we're seeing here is a result of its effect in this pathway. All right, let's see. There we go. All right, so for case one's conclusion, so ultimately we went ahead and before we even had a genetic diagnosis, we started him on IVIG. And because of his history of pneumonia, we ended up getting a CT scan that did demonstrate mild bronchial wall thickening and some bronchiectasis with mucus plugging. So for that, we started him on MES therapy. We also started him on some steroids and albuterol. He was continued on hydrocortisone replacement for his known adrenal deficiency. But ultimately, because we knew about his genetic diagnosis, he had close screening from the endocrine folks, and he ultimately manifested with growth hormone deficiency and had to be started on growth hormone replacement. And then more recently also started to manifest with the alopecia that's very characteristic of this genetic diagnosis. We had offered other genetic testing to the family members to see for sure if this was de novo or inherited, but they weren't really interested, and they've been a tough family for follow-up in general. But I will tell you, his variant was probably de novo just because with regard to penetrance in this specific genetic variation, it's pretty highly penetrant. So it's unlikely that his parents harbored the same variant and didn't manifest with disease, especially at their age. All right, so let's move on to case 2. So now we have an 8-year-old, healthy, full-term Caucasian male with a history of celiac disease, type 1 diabetes, who's now presenting to immunology clinic after being recently diagnosed with pancytopenias. He has no prior history of significant bacterial, viral, or fungal infections, and previously had normal newborn screening. His family history was notable for hypothyroidism in his dad and paternal grandmother. No other family history of recurrent infections or autoimmunity. He has two healthy full sisters and one healthy maternal half brother. No maternal history of spontaneous miscarriage and no known consanguinity. So because of that history, we went ahead and got a little bit more immunologic testing, particularly given the cytopenias that he had. And you can see that he does in fact have cytopenias, pancytopenias affecting pretty much all of his cell lines. You can't see it, but he did have a little bit of mild T-cell lymphopenia, normal B-cell phenotyping, normal Treg phenotyping. He did have some evidence of autoimmunity in his positive Coombs, normal bone marrow biopsy, did have a positive ANA, and did have more hypergammaglobulinemia, not as far as his humoral immune system, it looked to be relatively intact based on this testing. So let's go back to this question. Would you send genetic testing on this patient? What do you guys think?
I would say yes, because I have no idea what he has.
But you can talk about genetics. See cancer. So the reasons why we did is that he did have multiple autoimmune manifestations. If you remember going back to that early slide, 50% of those reasons of why I might consider genetic testing on a family or on a patient have to do with immune dysregulation. He also had early onset of disease, similar to our first patient, and then a family history of autoimmunity. Reasons maybe against it, he didn't have a concerning infectious history, his bone marrow was normal, he didn't have any evidence of humoral immune deficiency. So, based on the talk today, you enthusiastically sent this gene panel, in his case, that contained 429 different genes that are important for the immune function. And you wait the results, and 2 weeks later, you get that. And you get 8 variants of uncertain significance. So this is kind of where I really want to spend a little bit more time in talking, what do you do now, right? And so this is something that you're certainly going to see if you haven't yet. There are many reasons why we see variants of uncertain significance. So as I pointed out already, the more genes you're testing, the more likely you're going to see them. Reasons might be because we don't know how that variation affects the protein function. We might not also know, based on the literature that's been reported to date, association with risk on certain diseases. Does that segregate with the disease, etc.? The other thing that can certainly play a huge role in the likelihood of seeing VUSs are the ethnicity of your patient. As we'll talk about, a lot of the sort of normal populations that we reference in trying to decide is this normal variation versus pathogenic variation has to do with a compilation of a population that is predominantly Caucasian, right? So if you're seeing an ethnicity that's not represented in that population, you're going to be more likely that initial screen through that's done before you get the results back, they're going to get these variants and not know how significant they are. And so you'll get a long list of USs.
I need to.
No, you're fine.
That comes back to my original question. So how do we know what the normal genome is with all these different ethnicities, and who do we call the normal genome?
Hold that thought.
Right.
Right. So I wish I could tell you that you could ignore them, but unfortunately you can't. And we were just talking earlier. I'd say from my personal experience, when we do make a diagnosis, it's 50% of the time we get a result back and you have a likely pathogenic or a pathogenic variant, as in our first case. But the other 50% of the time, it was initially the US. So I'll tell you, this patient that I just presented, we diagnosed them with a genetic disease, and I'll tell you how we did it. You can't ignore them. The other thing to keep in mind is that our understanding can certainly change with time. So it certainly, even if 5 years have gone by, might warrant going back and taking another look, either at the variants that were identified or having genetic testing reanalyzed as well. And again, just like I showed you, it could certainly feel overwhelming at first, but I think what I want to show you is kind of what I do and how I tackle this, because I think it can be helpful when you start to see this in clinic. So this is what I do. It's by no means like the gold standard. It's just what I do in clinic and kind of how I think through this. And so we'll take this one step at a time, and I promise we will answer your question. So the first thing that I tend to do is I try to eliminate VUSs that aren't really consistent with the patient's clinical or laboratory phenotype. And the reason why I do this first is because you can quickly then narrow down this long list and get rid of several in order to prioritize where you want to spend your most time. There's a couple of things that you want to consider though. So number one is definitely consider the inheritance of the disease. So if you have an autosomal recessive disease that the gene might be associated with, but you only have one variant identified in that gene, then it's less likely that that might be the causal variant. Certainly you could miss things. Maybe there was a large deletion on the other allele. Maybe the other allele, there was an intronic variant that resulted in lack of protein translation. But again, we're trying to prioritize where we're going to spend our time, and this is one way to do it. The other thing to keep in mind, because there is certainly, I told you, variable expressivity in how a certain disease might manifest, is think through how that gene and or the protein that that gene makes, how it functions within your immune system. So what role does it play in the pathway, similar to how we talked about with NF2, to decide, could this be maybe a manifestation of a pathogenic variant in that gene? So these are some tools that I use. Believe it or not, Google is one of the first things I do. And this is kind of the search structure that I use down there. But the reason why I use first is because you can quickly, oftentimes, find papers that have either talked about the disease that that gene is associated with to get a better understanding of phenotype, but you can also, Google's fantastic at searching through supplementary tables, and sometimes you can find papers where that actual variant, or maybe a variant that affects that amino acid or the amino acid next to it, has been reported as associated with disease. And so it can be very helpful to do a quick search and find potential useful information. The other tool that I use, you guys probably use this as well and are very familiar with the Performian. It's another really quick, easy source to get a lot of information about gene function, protein function, where that is sort of expressed in tissue, and then phenotypes of disease that it's associated with. All right, going back to our framework, the next thing that I do is I ask myself, well, does anybody else in the family manifest with similar phenotype? And it's very, very helpful to get a pedigree in this situation. Certainly you can do this, but I will tell you I have the benefit of working with a genetics counselor who will oftentimes go in and get a really detailed family history. So the family history in this patient actually was very important, right? So the thyroid disease and things like that may certainly play a role. If you're looking at testing on family members, it can be helpful to see, do they carry that same variant that might be manifesting as disease in another family member? It's important when you do have sort of an autosomal recessive disease, or in our situation, we didn't have two variants in the same gene, but if you did, you want to make sure that you know how that variant, or those two variants in cis or trans, meaning are they on the same allele or on opposite alleles? Because when you're doing next-gen sequencing, you can't tell if you have two variants on the same allele that's coming up versus one on each allele. And so there's a variety of ways that you can do that. You can do a more long read sequencing, or you can again look at family members, because if the variant was inherited and mom has one and dad has the other, then you clearly know that it's inherited trans. And then I've said this a few times, but it's becoming useful. You have to consider both incomplete penetrance, so that is mom or dad that truly have the same genetic variant aren't manifesting with disease, either because of, again, just a variety of factors, epigenetic factors, things like that, versus maybe age-related factors. Maybe they just aren't, they have to because they're not old enough, as well as variable expressivity. So, the same genetic variant, I have actually a whole family that have the exact same genetic diagnosis, the same genetic variant, and they all manifested differently. And the mom's not even symptomatic. So it's important to keep that in mind. All right, this goes to your question. So now, the next thing that I usually do is I look at how likely are we to see this in healthy controls. And so the way we do that is, the reason why we do that is because, again, immunodeficiency is rare. So you would expect them, if you have something that's truly causing disease, that it not be represented in the general population. You can actually easily, usually what I do is anything that's 1%, amino allele frequency of 1% or greater, it's very, very unlikely to cause a rare disease. But you can actually calculate what your amino allele frequency should be for that gene, that variant, depending on what your prevalence of the disease is. So you can kind of calculate to get a sense of when you might want to exclude variants based on representation in the general population. And then the other things to consider when you're looking at normal, again, sort of these normal control populations is, what is the ethnicity of your patient? Because it might be rare in the overall population, but if you go and you look at the specific ethnicities, like in Ashkenazi Jews, for example, it is very common to see in Ashkenazi Jews, but in the total population, it's not as common, and your patient happens to be Ashkenazi Jew, then you know that that's less likely to be pathogenic, and maybe it's just a normal variation within that ethnicity. So that's important to take into consideration. And then the other thing that you can easily access is how common is that variant in homozygous in patients, because it's very unlikely if you have a variant that's frequently homozygous in patients that it's going to be causing disease. And so tools that we use, so this is a big one that we use a lot, it's Nomad. So this is a database that's been around for a while now. I think it's been around since at least 2013 or 14. But it now contains over 730,000 exomes, over 76,000 genomes of greater than 80 or 800,000 individuals. And it's basically a data dump. It's these people that are presumably healthy, and I use healthy in quotations because they're from, testing that was done on patients that have cardiovascular disease, or maybe they have malignancy, maybe they have GI disease, but they don't have immune deficiency. They do some things to sort of, again, try to purify it by getting rid of patients that manifested with severe disease early, so severe pediatric disease, as well as they try to eliminate first-degree relatives of patients that have known genetic diseases. But the reason why I put this in quotations as healthy is because we know one of the things on that list of when to send genetic testing is things like malignancy, right? Malignancy is very commonly associated with patients that have inborn errors of immunity, as well as inflammatory bowel disease, another very common one. So it's not perfect, but it's kind of what we use to get a sense of how common is this in the general population. And then the last bullet point that you can't see up there is, again, this is highly represented with people from Europe and America. Those are the two most common ethnicities that we see. And up there in the top right, you can kind of see the breakdown. They're trying to get better through their versions. The most recent version, version 4, the blue are European people. So it's very skewed by European people. So if you have somebody that's from East Asia, they're not as well represented in this database.
What does remaining mean?
Oh, it's a combination of like miscellaneous, not enough to fill. If they listed all of them, it would be too long. So like the rest of the ethnicities that make a very, very small subset of the population. All right, going back to our framework. So after you talk about does this variant exist in the general population, it's next helpful to ask, has it been associated with disease? Has it been reported as disease? And there's a couple of different things that you can do to answer that question. The one thing that you need to keep in mind when doing this are when you're reading the literature, when you're looking at reports of association with this variant and disease, it's important to understand what validation was done, what testing was done to say that that association was in fact valid. Does that make sense? So, a lot of times people were studying the literature all the time that might assume that this variant is causal for this disease, but they did not do their due diligence in actually showing mechanism for that truly causes disease, and it could be just an association. So a couple of tools to keep in mind. ClinVar is one of them. This is again a site that anybody can report information to. A lot of like Invitae, Prevention Genetics, they will dump their results into here, as well as any functional testing that has been done in follow-up. But again, it's important to keep in mind what testing was done and how that was validated as far as the determination of was it likely pathogenic, likely benign, things like that. Another site that's actually, it's very, very helpful, and it's only going to continue to get better because it's sort of still a work in progress. It's ClinGen. So these are curated, they're going gene by gene and curating different variants that are known to cause disease by experts. So we know that in fact what's published in here is true and valid. And they also have things in here that can be helpful. So like the likelihood, the penetrance of a certain gene, or where variants might where more pathogenic variants are likely to localize, or is it more likely to have if you have protein expression, like you're able to see protein, the likelihood that that protein is functional, stuff like that, it can be very helpful for. And then a final tool, you've probably heard of the CIS listserv. This is listserv. So you can also sign up for this, and this is just an easy way to sort of pull or send a shout out to experts around the world actually to see if they've seen this variant. A lot of times variants have been seen, they have been analyzed, and they have not been published. So this could be an easy way to see, has anybody ever seen this variant? Has any testing been done to validate this? Here's an example. So this was somebody that had posted about 2 variants in a patient that was presumably presenting with what seemed to be apposite, and they had 2 variants in pair. And so they posted a listserv, and you can't see it down at the bottom, but Dr. Leonakis, who's an expert in this disease over at the NIH, replied back his experience and actually was able to set up unsaid sample for him to validate whether or not that was functionally relevant or not. And that can be very helpful, especially if you don't have a diagnostic lab at your fingertips. All right, we're getting towards the end, I promise. The next is how might that variant affect the protein? And so there's a couple reasons why this can be important. So if you know the function of the protein and where the important domains are, and if you know where previously pathogenic variants have been described, that could certainly be helpful in determining where your variant lies, whether or not it might be an important area of that gene. Again, as we mentioned, non-shift and frame-shift variants are more likely to be pathogenic because you have disruption of that protein as a whole. A lot of those VUSs are going to be missense variants, so they're not, it's oftentimes more difficult. All right, and then tools that can be helpful for this. So UniProt is another site that you can go in and you can actually, it gives you structure of the protein, it gives you where the important domains are. You can see information within different species as well as those tissues where that protein is expressed. So very helpful. Another thing that I often do that's very helpful is if you just do a literature search, oftentimes you can find a patient cohort or a review. So this is a review of patients with CTLA haploinsufficiency. And right there in the paper, they have the schematic of the protein and important regions of where the exons are and where different known or previously presumed described pathogenic variants might fall. And so you might find that your variant is either that's affecting that same amino acid or right next to it that might raise your suspicion that this might actually be pathogenic. All right. This next part, I think, since I started doing this, has really changed quite a bit, and it's looking at in silico testing. So what computer algorithms, artificial intelligence, machine learning, what can they tell us as far as to help skew, is this more likely pathogenic over time? And there are, in fact, many in silico scoring systems that exist. They either they either look at variant location, like we've kind of talked about a little bit, amino acid change, what did it change to, as well as evolutionary conservation. As I mentioned, this has changed significantly, and there are now pretty awesome tools that are available at our fingertips that can help, although it does not take the place of functional testing. So the idea is you're trying to narrow that pool of VUSs so that you can really focus on which VUSs you truly ultimately need to do that true in vitro functional testing on. One last thing to say about that is that, as I mentioned, they usually look at different things, and it's important as you're looking at in silico tools that you understand what it's considering when it's giving you the readout that it's more likely pathogenic or benign. For example, if you're looking at conservation and both tools you're looking at are looking at conservation measures, it's not as helpful than if you're looking at one is showing you function, one is showing you conservation. So it can create bias if you're looking at 2 scoring systems that are looking at very similar things within that variant.
I don't know what that means.
So this can be really important for like meta predicting tools. So like, for example, some tools look at a variety of different species, and they look to see in this species, how often do we see this amino acid flipped? And if it's frequently not, if it frequently stays the same, that's leading you to say that it's not tolerant of variation, that it's more likely to be, problematic if there's variation there. But if you look at 2 scoring systems that are both looking at the same thing, that can create bias because you're looking at the exact same thing. And so like, you're just labeling, it's like looking at, I can't think of an example, but it's better to look at a couple of different scoring systems that incorporate a variety of different aspects of why that variant might be more likely to be pathogenic versus benign. Does that make sense? All right, so I'm not going to go through all of these. There is a laundry list of in silico tools, and I could have filled up probably 10 slides with them. But these are many of the ones that I use, but I thought it'd be more helpful because oftentimes I'm asked, well, what the heck do you, I mean, like, how do I know which one to use? The ones that I'm going to talk about 3 in particular that I use pretty much all the time. The first is the combined annotation-dependent depletion score, also known as CAD score. This tool, what it does is it actually uses machine learning and AI it ranks all of the possible variants within a genome. And based on a variety of different algorithms, it tries to predict and it ranks the ones that are more likely to be tolerated, to be observed in the normal population versus the ones that are more likely to be deleterious or not observed, what you call it, simulated. And I usually look at the ranked scale, can't see it there, but it's a score of either 1 to 99. And what it tells you is, for example, if the score is 10, it's telling you that it's in the top 10% of deleterious variants. If the score is 20, it's in the top 1%. If the score is 30, it's in the top 0.1%. So the higher the score, the more likely in that ranking that it's towards the side of being more likely to be deleterious. Does that make sense? So it's an easy tool to use. It's free. You just go to this website, you plug in your variant, and it spits out both a raw score and the ranked score. And I usually look at the ranked score, and that's what I'm talking about. The next one is this evolutionary model of variant effect, and this goes back to what we were just talking about. So they call this EVE, and what it does is it's a conservatory evolutionary computational model that again looks at over 140,000 different species to decide, how likely it is to be observed in a variety of different species. But again, if you see it a lot in a variety of different species that there's no genetic variation at this spot, it's more likely to be intolerant of variation. And so again, if you go to this website, it's free, you just type in your gene, and then it pulls up a heat map. So on the on the x-axis is the actual amino acid. So starting from 1 all the way to, it keeps going this way. And then on the the y-axis is the different the different amino acid change from your reference. And so if you scroll down, it gives you lots of information. So it gives you what it's calling pathogenic or benign or uncertain, depending on, the higher the number, closer to 1, the more likely it is to be pathogenic. If there's information available, it'll tell you the frequency and NOVAD. So it'll give you that information right there. And it even tells you if it's in ClinVar, if somebody's commented about that variant or that amino acid change in ClinVar, it'll bring that to your attention. So
Does that mean that people have already sequenced the genome in 140,000 species on Earth?
yeah,
Whoa.
yeah, it's pretty crazy. I mean, it's one of those things. So going back to the initial slide of just diagnosis of immunology, of the inborn areas of immunity, the next gen, the massively parallel high throughput sequencing that allows us to sequence quickly and very cheaply has really expanded all of the genetic information that we know, not just in humans, but in a variety of other different species. All right, the last one that I'll talk about is actually, again, it's a meta predictor. So what it does is it actually takes those tools that I talked about, right? Those in silico tools, it looks at 13 different of them. And it takes their output and combines it into one score. So these are actually 18 different scores that come up like 13 individual tools, and it puts it together in, again, a total score of 0 to 1, higher being more likely to be pathogenic. There's a really good paper published last year, I think, where it compared another meta predictor, and then several other in silico tools, and Revel outperformed all of the individual tools. Because it's taking more than just one aspect. But this goes into where I was talking about, because this is taking several different tools into one score, you have to be very careful, because if you're adding on other tools that are included in this score, you might create bias and just try to build this is pathogenic or benign when you're just using the same test over and over again. But as a meta predictor, using all these things, it can be very helpful. And again, it's free. The way this one works is you download, they've given you a Revel score for every possible variant, and then you download it in an Excel file and you can search for it. Don't worry, I'm not going to do that. All right. And then last but not least, we talked about it already, but functional testing. And so, how can you get a sense of what, this is the best, but often the hardest thing to do. You can look at maybe how it was functionally assessed, how variants in this gene were functionally assessed previously in PubMed, Omen, ClinGen, etc. You can consider the location of the protein and whether or not expression of that protein is more likely to be associated with, disease or not. So oftentimes you can assess for that protein by flow, for example. And if you know that it's a disease that if the protein is there, it's very likely to be functional. Keep in mind, oftentimes you can have a non-functional protein there as well. So you might detect the protein, but it's a non-functional protein. So it goes into just knowing, the caveats of that functional test you're doing. You can also knowing how it falls in the pathway. So again, an NF-kappa B pathway, You might look at stimulation through LPS to activate, or TNF alpha to activate the receptor, and then look for readouts. You could look at sort of that cascade, or in my example here, looking for the expression of CTLA gives you a way of evaluating for LRBA deficiency, which is upstream of CTLA4. All right. So this is where I was going to get to. This is the bonus tool that I'm going to give you that's going to make this a whole lot easier. And if you don't remember anything else other than like what I talked about, this is what I want you to go to. So this is Varsome. Varsome is a free tool. It has a paid option as well, and many of my my counterparts on the genetic side of things actually pay for the paid version. It gives you more information, which truthfully you don't necessarily need. But it gives you most of the information I gave you already is available in here in one place. I'll show you kind of how that looks like. So, but first let's go back to the case. So this was our list, again, 8 different variants. And so what I did here in this case is I just quickly looked at, again, disease inheritance. And because all of these variants, there weren't 2 variants in the same disease. So again, on first pass, I'm just trying to prioritize which variants I'm going to spend my most time with first. Prioritize my time on. I eliminated all the genes that were associated with only an autosomal recessive disease. They did not have a dominance, sort of association with disease. And so in doing that, I was able to eliminate all of those. I then looked at OMIM, and I looked at what does this phenotype of this genetic disease look like, and compared it to my patient. And in doing that, I eliminated 2 more. So we already narrowed down our list pretty drastically just in the first step or 2 of what I talked about. I looked at the first one on our list, which is STAT3, which you guys are probably very familiar with. And I looked at OMIM. And this is really pulled from OMIM. So STAT3 can cause loss of function. That's really hyper IgE, but it can also be associated with an autosomal dominant gain of function. And in the description, things stand out. Insulin-dependent diabetes, celiac disease, autoimmune hematologic disorders, anti-pain patient, hypothyroidism. This is starting to really build a case of, man, this sounds very so like my patient. And so I plugged it into Varsome. And so what I did here was I just tried to pull what the readout looks like. So you plug it in, and it gives you several different panels. And I copied here, which again, you can't see because of the box at the bottom, but what the ACMG criteria look like. So there were 3 different classifications that this variant met based on it not being in Nomad. So that was one of them actually, based on its publication in ClinVar, and one other thing that I can't remember there. But so ultimately it was still with those criteria classified as uncertain significance. So starting to build a case. So then what I did was I clicked on this tile, and I looked at in silico predictors. So remember we talked about, so I showed you one meta predictor, which is Rebel, and that's that line at the bottom. 0 to 1, right? Closer to 1 being more likely to be pathogenic. In this situation, Rebel wasn't very helpful, but there were several other meta predictors. So again, these are predictors that are taking several tools and putting into one score that were strongly making a case that this might actually be pathogenic. And then Varsome also lists, and there's more that I could fit on the slide, all of these individual tools as well. It doesn't automatically give you CAD, that's in the paid version, but you can go to that CAD site that I showed you and type it in. You can use the genetic sequence location information from Varsome and just copy and paste it in the CAD site, and it'll give you that score. But on this list, when it's covered up, Eve is on this list, Shift is on this list, Dan is on this. There's several different individual in silico tools that are just free here in one spot. And so again, it's starting to, what I'm using this in silico's tool testing for is to push me towards making a case that, man, this really sounds like this could be the reason for this patient. And so then you can either phone a friend, or I happen to have, there's one more thing. I looked and Googled, I searched literature, and this was a publication from a few years back of, again, various STAT3 gain of function variants that have been described in patients that have STAT3 gain of function. And what was really interesting here, this was kind of the clincher, is that the variant in my patient had a variant, and if you remember the reading frame, right? The three nucleotides that then equate the amino acid. This patient in this paper, this is the same amino acid that my patient had a variant in that changed the amino acid. It's just that it changed it to a different amino acid than what was published in this paper. So that same amino acid site resulted in disease, a variant in that site resulted in disease in a cohort of patients that had STAT3 gain of function. So now, I mean, you're really like, this has got to be the answer. And then, so in the end, I have one of the most awesome diagnostic immunologists in the world. Roshni Abraham is our diagnostic, she's the director of our diagnostic immunology lab. And so she was able to do an in vitro assay to look at STAT3 function and STAT3 activity. And so in our patient, which was this variant, it was increased over wild type. So made the diagnosis of STAT3 gain of function in this patient. Then the other important thing is when you do this, if you do this, it's important to go back and let whoever did the genetic testing know, because then they'll go update that in ClinVar, and they will utilize that, because if they see it again, if somebody else, this variant hadn't been described before, but if you let them know, that can be helpful down the line for somebody else. And then certainly, if you can, publish it too.
How does that variant lead to those clinical features?
So a variety of different ways. So one way is that overactivation of STAT3 ultimately leads to an inhibition of regulatory T cell function. So you have basically a skewing of your immune system down like the STAT3 pathway. And you have, if you look at their oftentimes they'll have regulatory T cell dysfunction, they'll have low Tregs. There's a variety of different ways, but it's in a dysregulated fashion that it manifests disease. But a lot of the autoimmunity is thought to be, again, because of that skewing, and then some partially with the regulatory T cell piece as well. So takeaways from the talk. So number one, making genetic diagnosis can have a significant impact on diagnosis and management of patients with families with inborn errors of immunity. I think we can all feel comfortable in saying that. It's important to consider genetic testing in patients that have these features that might be more likely to be an underlying inborn error, which we went through. VUSs, you're going to see them, and unfortunately, you can't ignore them. Hopefully, and if you just kind of take things systematically and one at a time, you can at least start to prioritize which variants might be more likely to cause disease, and then you can always phone a friend and utilizing the various tools that I showed you. So with that, I'm happy to take questions, but hopefully that was helpful. Anybody have any questions?
How do you describe the difference between ClinVar and ClinGen?
ClinVar is just a public database that anybody can go in and input the variant that they found and like what they did to say that it was pathogenic or benign and what they think it is. So they call it pathogenic or benign. But anybody, you can go in there and you can put it in. Anybody can. ClinGen, there are actual experts, and you can actually sign up to be volunteer time to do this. They teach you, they train you how to do this. Actually go in and systematically curate each like different variants, and they curate each gene to provide information as far as what aspects are important to consider when evaluating variants in the gene. But then they've also started to actually curate individual variants that have been identified. And you can go in there and they'll even link to papers where they've been described and things like that. So what's in ClinGen is much more reliable than what's in ClinVar. That makes sense.
Okay. Yes. Thank you.
Who supports all these sites that are free? How does that come to?
Some of them are government. So ClinGen is government, ClinVar is government.
Or
Some of them are I want to say, I think CAD scoring is it's George Washington University, I want to say. So some of them, one of them, so Stanford is another one that has tools that you can utilize. A lot of academic institutions will have them, and it's based on, so if you go to the site, like let's say you go to the CAD score site, you can click on About, and it tells you how it was developed, and it links to publications of when it was first published as how it might be useful in silico testing of different variants and how it was validated. And so that's where you can get a lot of the information. So a lot of it is, again, it's not for some of the in silico testing, it's not like there's not ongoing work that needs to be done, right? It's a machine that, for example, Level, it already, or not Eve, for example, it already went through the entire list of possible variants that could occur. And for most of them, it already inputted input out what it thought was going to be relevant for that variant, for the amino acid change. So there's not ongoing, aside from updating and adding in new updated information as we learn more, it's not like somebody sitting behind a computer and every time you plug it in, they're like, for example.
Sounds to me like with the advances in AI, this is gonna become amazing.
I will tell you, when I first gave a lecture on sort of this theme was maybe 6 years, 5, 6 years ago. And at that time, I put very little emphasis in silico testing. And it's surprisingly, even in 5 years, how far AI has been able, machine learning and deep learning. So machine learning is where you're actually helping the computer tell it what is right, what is pathogenic, what is not. Where deep learning is what's used in many of these, where you basically input all this data. You don't need a human to necessarily go in and tell the machine, and the machine learns on its own. And so like Eve is unsupervised machine learning. So it basically looks at these over 140,000 different sequences of different species, and it uses that information to predict what is more likely to be tolerated and not. And based on you just input this data and you put in the algorithm, and this neural network will like learn on its own.
It's a new world.
Could you speak a bit more about, what's the process of like making something officially pathogenic? I mean, it's all you publish, for example, of course.
Yes. So
Repeat her question. I'm not sure the outside audience heard.
so she's asking, how do you make it official that something is truly pathogenic versus not? So one is, again, what's been reported in the literature. So the reason why it's important to go back and let the testing know is because you did maybe functional validation on that then is taken in consideration when you're going to the AMC, the American College of Medical Genetics and Genomic criteria. If you go back to that thing, you can input like, okay, we did functional testing and that's there. It's actually very strong evidence that is pathogenic, and then you only need one other like pathogenic, like strong evidence thing, or like a couple things to then specify that as pathogenic. So part of it is utilizing that table that I showed you. But as far as the report that you get back, listed pathogenic, likely pathogenic, benign, likely benign, it is based on what they have seen in the literature, what they know about that variant, and input it into either that algorithm that I showed you, or like in Vitae, they have a lot of different of their own sort of variant interpretation software that they use in order to help, again, push those USs a little bit more one way or the other. And next year, they're actually going to be, as I mentioned, they're going to be dating that whole algorithm. And the whole reason for that is those variants of uncertain significance. I think what they're going to do is actually there's going to be a bigger role for in silico testing and how to utilize that, because more studies have been published, more papers have been published to show that in that 2015 criteria, the best you can do, all in silico tests are considered the same. But if you have two that call it pathogenic or two that call it benign, you can skew your test one way or the other by a little bit. But there are some scoring systems like Rebel, for example, that take in consideration all these various tools into one outcome that spits it out. And that outcome has a much stronger weight than what that table gives it credit for. So I think in the next iteration, they're going to start to pick that apart as far as, maybe offering different weights depending on which tools you're using in an effort to narrow that US space. Because if you look at all variants that are reported out, like when you get variants back, 50% of them are USs. So that's why this piece is so important, and why the in silico piece is so important to help narrow that down, so that you're putting your time where it's being most beneficial.
Where do you see this going in 10 years then? Do you think, yeah,
I mean, I truly think like what you hit on when we're talking about, I think that the thing that's gonna have the biggest piece is going to be probably machine learning, artificial intelligence, the use of computers to help determine based on predictors the likelihood that a variant is pathogenic or not. I think the other thing that's going to be, I think, hugely different in 10 years is going to be our understanding of what's in the intronic space, right? And how to interpret intronic variants. So like I said, we know most of your genome is actually not even coding. It's in the introns that are spliced out when you're making the protein. And I think it's very naive of us, and I think when we first sequenced, human DNA, I think the attitude was, now we know, now we can classify all of these diseases, and I think what we learned is what we don't know. And so starting to shed more light on, we don't know what other genetic material does, all of it. And clearly it's got to be important, And so why is it that, so the family I talked about actually is not this patient's family, but it's a cohort that has some 3 gain of function. And the mom has a pathogenic variant we published it. She has 2 siblings, older siblings. One almost had a heart lung transplant, severe humoral immunodeficiency, very sick. His sister, similarly, not quite as severe as him, but again, has pretty significant disease. And then there are a couple of family members. Mom is completely asymptomatic. It's the same variant. There are a couple other half siblings that again have the same exact variant, no disease. Mom's brother, so uncle of this family, has the same variant, no disease. His offspring, his daughter, manifested about, I think, 2 years of age with very early onset inflammatory bowel disease, very characteristic of STAT3 gain of function. So, I mean, why is that? There are all these other factors that are going, it's not just the code, right? And so I think understanding of that other, and that's also, I think, goes into the interpretation piece. You asked me, like, why is it only 30%? It becomes very difficult when you're analyzing that family. So if you didn't know about STAT3 gain of function and you were analyzing that family, it's an autosomal dominant disease, and you have several family members that manifest no disease whatsoever. It's very difficult to show, like, why would you think that why is that gene the one that would cause disease when you have some that have that don't have disease and some that don't? So, or some that do. So it can be very difficult to, interpret which pathogenic or what variant is truly pathogenic and disease causing.
This isn't a question, it's more a statement. My concern is, can our society afford this? Because here we're dealing with very rare diseases and spending, I presume, massive amounts of money. Even if the things are free, someone's paying the bill, the federal government or academia. While in certain settings, we don't take good care of young children in inner city, poor neighborhoods, where we put our priorities. So this is absolutely fantastic, but are our priorities in the right place?
Yeah, I think that's an excellent point. I think that the benefit of this will only get greater as we go on, right? The more we know, the more we can then apply that knowledge. But
I'm not being critical.
No. And I will say again, certainly understanding played a role, but really probably the big reason for the exponential increase in genetic not just with immunology, new inborn areas of immunity, but in many different areas of medicine, like the rapid increase in novel genetic diagnoses. The big factor is cost. It's gotten so much cheaper to sequence now. And so people can do it. I mean, you can get Invitae, going through insurance. You could just order the test online. They send you a kit, $250. I mean, it's still a lot of money.
Yeah.
But that's not, in the grand scheme of things, if you could find something out that would then help, down the road for, either screening or family planning or all these other factors, that can be huge as far as saving money.
Are other specialties of medicine as advanced, or is this unique to immunology?
Oh no! So if you go into ClinGen, there are a variety. It expands like many areas of medicine. And there are different expert panels that are curating variants in genes that are important for their disease or for their subspecialty. So this is impacting, I think why this has made such a big role in our disease is one, is we're interested about it. Two, if we can be honest, like our testing for the immune system is very poor, right? If you think about it, we can look at antibodies, we can look at responses to vaccine, we can phenotype, and we can look at, T cell numbers and things like that. But when you're starting to, there are a lot of different, there's a big overlap in how inborn errors can present. If you just look at this patient, how would you know to analyze this specific immunologic pathway? Because that pathway, a variant in that pathway could present like this other pathway, could present like this other pathway. So that initial screening test, it gives you little pieces to and sometimes you can make the diagnosis, like skin does not require a genetic diagnosis, but oftentimes the genetic diagnosis is suspected, and then the functional testing is done to prove that that's the answer. It's kind of reverse.
Could we, or did we use all this technology during COVID to, we just looked at antibody responses in an old fashioned way. Could we, or should we have said, when we use this vaccine or this vaccine, we got this more elegant outcome?
So, there were a variety of different inborn errors of immunity that were identified, or I would say immunologic susceptibilities to having more severe COVID that were identified. Another piece that came out in COVID was the auto antibody piece, like especially in older adult males, the frequency of auto antibodies to interferon, for example, was a huge correlative to severe disease. So it's clearly multifactorial. And how much of it is truly genetic and only genetic? It's hard to say. Certainly there's a good amount that's there. But yeah, some of this certainly helped with that. But the other thing that I oftentimes will share with patients now from the COVID experience, because how many times do you see patients in clinic who their kid has had 10, every month or twice a month, they get an upper respiratory tract illness. They maybe had recurrent ear infection, but have never had pneumonia, never had all this stuff, and the family wants an answer. Why is my kid so sick? Why are they getting sick? And we know that it's not like a tangible, they don't have a true humoral deficiency, and we oftentimes test just, to reassure. But oftentimes what I'll say to them is while there was a lot of bad that came from the pandemic, we also learned a lot. And one of those pieces was look at the variability. We have one person that has COVID that truly has an infection, completely asymptomatic. You have another person that had, was symptomatic for 2 weeks. Neither of them had a true defined underlying inborn error of immunity. And it's just all these other, unquantifiable factors that go into the manifestation of the disease that we still don't fully understand.
Well, that was, for me, who knew nothing about this, a brilliant talk. Oh, thank you so much. How many people did we have listening in the outside world? 27. All people learned that we're not in this room. Well, thank you.